السلام عليكم أعزائي الطلبة. اليوم راح نتكلم على موضوع جدا حيوي، وهو تعرفون الجائحة اللي حدثت خلال العامين الحالي واللي قبله، جائحة كوفيد، والصراع اللي بدأ بمختلف الشركات للحصول على حماية ضد هذا الفيروس، وهو إنتاج فاكسين اللي هو كوفيد 19 فاكسين. تعرفون حاليا الفاكسينز اللي توفر في العراق هو 3 أنواع. 2 من عندهم هم ماسنجر RNA فاكسين اللي هو فايزر وأسترازينيكا، وواحد من عندهم inactivated vaccines. فراح نتطرق إلى دور الاميون ريسبونس against infection, how the vaccine will elicit the immune response against the COVID-19 virus. So the COVID-19 pandemic has infected millions of people with no clear signs of abatement owing to the high prevalence, long incubation period, and lack of established treatment or vaccine. Also, we can add there is various host for the reservoir of the infection, like animals, like bats, and so on. Although physical distancing, quarantine, and isolation were effective in limiting the number of people becoming infected during the pandemic in a short term, the absence of immunity in the population left them susceptible to further waves of COVID-19 infection. احنا من نلبس الكمامات، يعني كل البيرسونال بروتكتيف ايكوبمنت، هذا تعطينا يعني وقاية آنية. هاي الوقاية الآنية راح تختفي عند إزالة الايكوبمنت اللي احنا ارتديناهم. لذلك هذا ما راح يعطينا مناعة دائما، هذا راح يعطينا فقط protection against of getting infection. So there should be a way to give an immune response against to COVID-19 infection. Health care worker, older people, their age range more than 60 years, and those of underlying health condition like diabetes, hypertension, ischemic heart disease are at particularly high risk of developing infection. The shortage of an effective treatment for COVID-19 has led to a quick action in the development of potential vaccines against the disease. Consensus among experts is that only the effective COVID-19 vaccine will end the pandemic. There is no way to eradicate the disease except the development of highly effective vaccine. So various vaccine platforms and strategies in parallel is essential, because little is known of the nature of the protective immune response to COVID-19 till now, and which vaccine strategies will be the most successful is unclear. So vaccines are the most promising solution to mitigate new viral strain. The genome sequences and the protein structure of the COVID-19 novel coronavirus were made available in record time, especially the genome sequences and their protein, spike protein, which is the main target for the entrance of the virus to the susceptible cells. And this allows the development of inactivated or attenuated viral vaccines, along with the subunit vaccines for prophylaxis and treatment. Since much of the pathology of COVID-19 is outside the airway due to systemic viral infection, a vaccine that elicits immunoglobulin G antibodies could protect against infection from systemic circulation of the virus. IgG antibodies opsonize the target antigen, presenting the opsonized product to the phagocyte while also activating the complement system. In these ways, the immunoglobulins will accomplish the eradication of the viral antigen or virally infected cells. Another hallmark of vaccine development is T cell involvement. And differences in T cell response can influence generation of high affinity and neutralizing antibodies, as well as elimination of the infected cells. Immune memory and generation of high affinity class switched antibodies are highly dependent on T cells and normally do not develop without proper T cell involvement. So, in this way, there is a coordination between both extremes of the immune system, of the adaptive immune system, mainly the B and T cells involvement. تعرفون دور of T helper cells بالتفاصيل المحاضرات السابقة how the T helper cells will coordinate the immune response, whether it will cause cytotoxic T cell stimulation or B cells stimulation. خلال هالفترة الصارمة ومنذ بدء الجائحة شفنا مختلف شركات الأدوية أعطت دور كبير في تصنيع لقاح against COVID-19. So there are over 100 vaccines being developed throughout the world, and the race to be the first effective vaccine has fueled the rapid development of both preclinical and currently utilized vaccine approaches. This table summarizes the clinical trial and the trial that have been completed by companies in the vaccination effort against COVID-19 virus. The table shows the company, the vaccine type, the vaccine name, how the vaccine will work, and what is the situation at that time. The main vaccines, the Mrna Vaccine, which depend on the technology of the messenger RNA technology, the BioNTech Visor also give the same principle. Both of them will code for the messenger RNA of spike protein and expression of this spike protein on the surface of the target cells to be recognized by the immune system. Another type of vaccine is the University of Oxford vaccine AstraZeneca, which is the non-replicating viral vector, and CanSino Biology's non-replicating viral vectors. Also, the Sinovac vaccine, which is the available now in the country. These figures shows an overview of the different vaccine platforms in development against COVID-19 virus. The first one is a whole inactivated virus vaccine. The best example is Sinovac and Sinopharm vaccine. The second one is live attenuated COVID-19 vaccine, still in a preclinical trial. This type of vaccine carry a risk of conversion to IL virus, so there is limited experience in this type of vaccine within this time. Could be a further trial in this vaccine to prove its efficacy against COVID-19 vaccine. Another type is the subunit vaccine, the weakest type of vaccine development till now. Or another type is the virus-like particle vaccine, the new generation vaccine, example viral vector. The best example is the AstraZeneca virus vaccine, which the messenger RNA of the COVID-19 vaccine incorporated into adenovirus of monkey origin. The another type is the DNA vaccine, and the last one is the antigen presenting cell still in a clinical trial. So there is different protocols in the development of vaccine against COVID-19 for emergency use during this pandemic. Now we will talk about the three different viral vaccine against COVID-19. The first one is the Visor Vaccine, which is the messenger RNA vaccine produced against spike protein. This give a protection of about 95% against COVID-19 after the full vaccination program. The second vaccine is the AstraZeneca vaccine, which produced also as a messenger RNA. This type of vaccine is incorporated with the adenovirus vector as a carrier for messenger RNA that encode for spike protein. Also carry high efficacy after full vaccination program. It carry about 95% protection. Both vaccines, Pfizer and AstraZeneca, carry a high efficacy against different serotypes of COVID-19 vaccine, even give a protection against the 3-variant vaccine that is raised during outbreak in India. Compared with an inactivated SARS-CoV-19 vaccine, CoronaVac or Sinovac vaccine is an inactivated vaccine candidate against COVID-19 that has shown a good immunogenicity in phase trial in mice, rat, and non-human primate with vaccine-induced neutralizing antibody to COVID-19 virus, which could neutralize 10 representative strains of COVID-19 virus. Moreover, the result indicated that CoronaVac provided a partial or complete protection in McCoys from severe interstitial pneumonia after SARS-CoV-19 2 challenges without observable antibody-dependent enhancement of infection, which support progression to a clinical trial in a human. And this happened where this type of vaccine is distributed in different type of countries, include Gulf countries, United Arab Emirates, and in Turkey and Egypt, and different type of South American countries. And lastly, this vaccine were available in Iraq for clinical vaccination program. Compared with other COVID-19 vaccine candidates, such as virus vector or DNA or RNA vaccine, the occurrence of fever after vaccination with CoronaVac is relatively low. Now, in the next slides, we will see different type of vaccine, how to be manufactured, and mode of action of type of vaccine, and how the immune system will trigger triggered against infection with COVID-19 vaccine.
All COVID-19 vaccines being developed aim to produce immunity to the SARS-CoV-2 virus by stimulating an immune response to an antigen, usually the characteristic spike protein found on the surface of the virus. Some traditional vaccines achieve this by modifying the virus to weaken or disable it, so that when introduced to the body, an immune response can be generated to the antigen, without the virus causing disease. When the immune system comes into contact with the weakened virus, its defenses, such as antibodies and T cells, attack the virus or infected cells. In the process, specialized memory cells take note of the specific antigen and prime the immune system to produce cells and antibodies that will quickly target these proteins. So the next time that person is exposed to the same virus, the immune system is ready to fight it off. Instead of using the whole virus, another way to trigger immunity involves using just fragments of it, like the spike proteins. These subunit vaccines have the advantage of being relatively easy and cheap to produce, and are incapable of causing disease because these fragments are not able to infect host cells. However, they are less likely to be recognized by immune cells aimed at attacking infected cells, which means they may trigger a weaker immune response. Because of this, subunit vaccines often include chemical agents called adjuvants, which are designed to stimulate a stronger immune response, and booster shots may also be required. Not all vaccines are designed to introduce antigens to the body. Some work by using cells in a patient's body to produce the antigens themselves. Examples include viral vector vaccines and mRNA vaccines. In both cases, the goal is to get a short fragment of genetic code taken from the target pathogen, in this case the SARS-CoV-2 virus that causes COVID-19, into the patient's cells. By hijacking the cellular mechanisms, these kinds of vaccines mimic the way viruses normally reproduce during natural infection. But rather than creating copies of the virus, the cells only produce large amounts of antigen that then usually trigger a strong immune response. Viral vector vaccines achieve this by inserting the genetic code for the antigen into a harmless virus, which effectively acts like a delivery system to get the code into the cells without causing disease. Vector-based vaccines can be complex to develop, but they can trigger strong immune responses without the need for adjuvants. And in theory, one type of vector can be used to deliver code for a range of different antigens, which can speed up vaccine development. Nucleic acid vaccines, like mRNA and DNA vaccines, also involve inserting genetic code into cells to produce antigens. But instead of using viruses to deliver the code, these vaccines take a more direct approach that involves inserting the code directly into cells, either by attaching it to a molecule or by forcing it into cells using a gene gun. These vaccines can be quick and cheap to develop, but they are a relatively new technology. With hundreds of COVID-19 vaccines now in development, it's likely that a mixture of different approaches will be needed in order to stop the global spread of this coronavirus and end the pandemic.
سينوفارم. لقاح سينوفارم مصنوع من فيروسات كورونا معطلة، يعمل عن طريق تعليم الجهاز المناعي صنع أجسام مضادة ضد فيروس كورونا، حيث ترتبط الأجسام المضادة بالبروتينات الفيروسية الشوكية الموجودة على سطحه. كيفية التصنيع. لإنشاء لقاح سينوفارم حصل باحثو معهد بكين على 3 أنواع من الفيروس التاجي من المرضى في المستشفيات الصينية. اختاروا أحد المتغيرات لأنه كان قادراً على التكاثر بسرعة. تعطيل وقتل الفيروس. بمجرد أن أنتج الباحثون مخزونات كبيرة من فيروسات كورونا، قاموا بصبها بمادة كيميائية تسمى بيتا بروبيولاكتون، حيث يقوم المركب بتعطيل فيروسات كورونا عن طريق الارتباط بجيناتها، وبذلك تفقد الفيروسات التاجية المعطلة القدرة على التكاثر، لكن بروتيناتها بما في ذلك الشوكية تظل سليمة. ثم قام الباحثون بسحب الفيروسات المعطلة وخلطها بكميات ضئيلة من مركب قائم على الألومنيوم يسمى مادة مساعدة، لتحفيز جهاز المناعة على تعزيز استجابته للقاح. دفع الاستجابة المناعية. نظراً لأن فيروسات كورونا في لقاح سينوفارم ميتة، فيمكن حقنها في الذراع دون أن يصاب الشخص بكوفيد-19. فبمجرد دخول الفيروس الميت الجسم، يتم ابتلاع بعض الفيروسات المعطلة بواسطة نوع من الخلايا المناعية يسمى خلية مقدمة للمستضد. تمزق الخلية فيروس كورونا وتخرج بعض شظاياها على سطحها. هنا يمكن أن تكتشف الخلية التائية T، الجزء الموجود على السطح. إذا كان الجزء مناسباً لأحد بروتينات سطح الخلية التائية، فيتم تنشيط الخلية التائية، ويمكن أن تساعد في تجنيد الخلايا المناعية الأخرى للاستجابة للقاح. صنع الأجسام المضادة. الخلايا البائية B نوع آخر من الخلايا المناعية قد تواجه أيضاً فيروس كورونا المعطل. تحتوي الخلايا B على مجموعة كبيرة ومتنوعة الأشكال من البروتينات السطحية، وقد يكون لبعضها الشكل الصحيح للالتصاق بفيروس كورونا. عندما تغلق الخلية B يمكنها سحب جزء أو كل الفيروس داخلها وإخراج شظايا فيروس كورونا على سطحها. يمكن للخلية التائية المساعدة التي يتم تنشيطها ضد فيروس كورونا أن تلتصق بالجزء نفسه. عندما يحدث ذلك يتم تنشيط الخلية B أيضاً. عندها تتكاثر وتخرج الأجسام المضادة التي لها نفس شكل بروتينات سطحها. إيقاف الفيروس. بمجرد التطعيم بلقاح سينوفارم يمكن لجهاز المناعة الاستجابة لعدوى فيروسات كورونا الحية. تنتج الخلايا البائية B أجساما مضادة تلتصق بالغازات. يمكن للأجسام المضادة التي تستهدف البروتين الشوكي على سطح الفيروس أن تمنع الفيروس من دخول الخلايا. تذكر الفيروس. الذاكرة المناعية. أظهرت التجارب السريرية لشركة سينوفارم أن لقاح سينوفارم يمكن أن يحمي الأشخاص من كوفيد-19، لكن لا أحد يستطيع حتى الآن تحديد مدة استمرار هذه الحماية. من الممكن أن ينخفض مستوى الأجسام المضادة على مدى شهور، لكن الجهاز المناعي يحتوي أيضا على خلايا خاصة تسمى خلايا الذاكرة B التي قد تحتفظ بمعلومات حول فيروس كورونا لسنوات أو حتى لعقود.
Viral infection, the virus attaches itself to a human cell using its spikes. Following entry to the cell, it uses the host cell's machinery to replicate, producing viral proteins and genetic material. These are then assembled into new viral particles, which are released as the host cell dies. New viral particles again infect more cells, destroying body tissues, producing symptoms. Infected cells alert the immune system by displaying pieces of viral proteins on their surface. By doing so, infected cells are said to present the viral antigen to certain immune cells, namely cytotoxic T cells, and thus activate them. At the same time, debris of dead cells and viral particles themselves are picked up by so-called professional antigen-presenting cells, of which dendritic cells are most effective. Dendritic cells patrol body tissues, continuously sampling their environment for intruders. After capturing the antigen, dendritic cells quickly leave the tissue for the nearest lymph node, where they present the antigen to another group of immune cells known as helper T cells. Viral particles also activate B cells in lymph nodes. These groups of immune cells work together to mount two types of immunity specific to the viral antigen: cell-mediated immunity and antibody-mediated immunity. The process, however, may take up to a couple of weeks, during which time the person is sick and the virus will have destroyed a vast number of cells. Vaccines deliver viral antigens to trigger immune responses without causing the disease. The events of a vaccine-induced immune response are similar to that induced by a natural infection, although some types of vaccines may induce only antibody-mediated immunity and may therefore be less effective. The immune process can sometimes produce symptoms similar to a mild infection, even though there is none. As the lymph nodes near the injection site start producing antibodies, they may become swollen and tender for a few days. These are signs that the vaccine is working. Many existing vaccines contain a weakened or inactivated virus. Because the whole virus is used, these vaccines require extensive safety testing. Attenuated vaccines may still cause disease in people with compromised immune systems. Inactivated vaccines only induce antibody-mediated immunity. Subunit vaccines contain only part of the virus, usually a spike protein. These vaccines cannot cause disease, but they may not be seen as a threat to the immune system and therefore may not elicit the desired immune response. For this reason, certain substances, called adjuvants, are usually added to stimulate the antigen-presenting cells to pick up the vaccine. A subunit vaccine may also consist of empty virus shells without genetic material. Having a typical size and shape of a pathogen, these vaccines may not require adjuvants to be perceived as danger, but they can be difficult to produce. Nucleic acid vaccines contain genetic information for making the viral antigen instead of the antigen itself. DNA vaccines introduce viral DNA into the cell's nucleus, where it is transcribed into mRNA. The mRNA is translated into viral spike protein in the cytoplasm. The protein is then displayed on the cell's surface, just like with other types of vaccines. Naked DNA vaccines require a special delivery method to reach the cell's nucleus. Alternatively, a harmless, unrelated virus may be used as a vehicle to deliver the DNA. In this case, the vaccine is also known as a viral vector vaccine. For example, the Oxford AstraZeneca COVID-19 vaccine uses a chimpanzee adenovirus as a vector. The adenoviral genome is modified to remove viral genes, and the coronavirus spike gene is added. This way, the viral vector cannot replicate or cause disease, but it acts as a vehicle to deliver the DNA. The reason a non-human adenovirus is used is because most people may have been exposed to human adenovirus and have immunity against it. The immunity could destroy the vehicle before it can deliver the DNA and thus blunt the vaccine's effectiveness. DNA vaccines have raised concerns about the possibility of viral DNA integration into human genome. However, studies in animal models have shown that integration frequency is well below the frequency of natural, spontaneous gene mutations. mRNA vaccines introduce mRNA that contains information for making the viral protein. mRNA molecules are delivered within a lipid covering that will fuse with the cell membrane. Once inside the cytoplasm, the mRNA is translated into viral antigen, which is then displayed on the cell surface. Unlike DNA vaccines, mRNA vaccines are extremely unlikely to integrate into human genome, but they are fragile and require uninterrupted cold chain for transportation and storage.